My name is Rakesh Karmacharya. I wear two different hats. I'm a scientist at the Broad Institute where I with the chemical biology program. And I also am a psychiatrist and I see patients with schizophrenia and bipolar disorder at McLean Hospital. These are illnesses that start in sort of around adolescence and early adulthood and are debilitating and chronic, and patients really are non-functional most of the time, and they and their families suffer really for their whole lives. At the same time, our knowledge of these illnesses was so rudimentary. While it was fascinating and to learn about different psychological theories, the biology of it was so not understood. So one of the difficulties for us in psychiatry is trying to get the tissue material that we're interested in studying. In 2006 and 2007, Shinya Yamanaka, a scientist in Japan, found a way to reprogram any somatic cell, in his case it was a fibroblast, into what's called an induced pluripotent stem cell. So from this we can actually differentiate those embryonic stem cell-like cells, the iPS cells, into neuronal cells. So we actually then have live neuronal cultures that we can study in the lab that have the same genetic makeup as the patients. So this is a picture of one of those cultures after 2 months of differentiation. So what you see labeled in green are the neurons, and red are astrocytes, and the blue color just marks all the nuclei for the different cells. The astounding thing is this is a neuronal culture that started out as a skin cell. I think it's all of the above. On the one hand, I think to understand the biology of it is fascinating, but at the same time, right now, we have no way of testing patients. So if there's a way where we can clearly distinguish what kind of features are seen with schizophrenia versus bipolar versus controls, it would also make the diagnostic conundrum much easier. I think in some ways, the big goal for us is if we can find a robust disease signature, then to do high throughput screens of small molecules that will correct or modulate that process. This will be a way of trying to find some leads towards small molecules that may be useful as we look forward for new ways of trying to treat those illnesses.